All right, so this is going to be the second lesson for macromolecules and one of the final lessons in our unit 1. So for this lesson, I'm going to describe the properties of monomers and bonds that connect monomers in biological macromolecules, as well as structure and function of the biological macromolecules. So quick reminder of what a monomer and polymer is. Monomer literally means one unit. Or a monomer, a good example I'll use will be Lego bricks. A monomer would be a single Lego brick, but a polymer, where poly means many, would be a connection of a bunch of Lego bricks put together. So here's another relationship showing monomers to polymers to macromolecules. At the bottom, you can see an example that's a protein chain, where the smallest unit, the monomer, is called an amino acid. When we combine amino acids, we have what's called a peptide chain or a polypeptide chain. And well, in a large way, you can see there's a macromolecule, which is the polypeptide chain, which would be the protein. Okay, macromolecules, macro simply means big, molecule, group of atoms, so it's a big molecule. There are 4 major types of macromolecules we're going to discuss in this year of AP Biology: carbohydrates, nucleic acids, proteins, and lipids. So the different monomers of macromolecules are listed here. You can also see that we've used kind of the shortcut with the elements. So the easy way to recognize, for instance, let's say a nucleic acid is down here at the bottom, where we would be carbon, C, hydrogen, nitrogen, oxygen, phosphorus, and sometimes sulfur. The monomer would be a nucleotide, and examples of it would be DNA and RNA. This chart right here is a useful tool to use, and knowing the elements as well as different monomers and examples will be very useful. All right, the first one we're going to talk about is carbohydrates. So carbohydrates, like listed on the table, is made up of carbon, hydrogen, and oxygen. Examples include sugars and polymers of sugars. We call monomers of sugars monosaccharides, and a good example would be glucose. Carbohydrate polymers are called polysaccharides. They're made up of more than 2 monosaccharides. Carbs are used by the cells of the body to produce very short-term energy, and in plants, for instance, like cellulose, to help create structures. Monosaccharides tend to have a chemical formula with ratios of CH2O, so 1 carbon to every 2 hydrogen to every 1 oxygen. Glucose is a great example of that with 6 carbon, so it has twice as many hydrogen and an equal amount of oxygen, which it does. It has 6 carbon, so it should have 12 hydrogen, and it does. And it should have 6 oxygen, and it does. C6H12O6. Another term for CH2O is a simplified formula or an empirical formula. Glucose is the most common monosaccharide. The function of a monosaccharide is that it serves as major fuel for cells and as raw material for building some molecules. Examples of monosaccharides include glucose, fructose, and dextrose, where some of these, such as glucose and fructose, have the exact same formula, just different groups. All right, here's a great example of something we're going to talk about in a second. This is called dehydration synthesis, and this is showing a compound for glucose and fructose being broken, and then we have another example down here, the opposite, where it is being created. All right, here's an example of polysaccharide. First, we have disaccharide, which is when dehydration synthesis, shown right here, where the oxygen bonds the 2 molecules, joins 2 monosaccharides. And we have a polysaccharide, which is a polymer of sugars forming 2 or more. This has large storage and structural roles, especially in plants. It's made of 3 or more monosaccharides. So examples of that include things like starches, which would be plant version of stored energy, glycogen, animal version of stored energy, cellulose, and chitin, which are 2 structures that provide support: cellulose in plants and chitin in animals. Our second one is lipids. So lipids are nonpolar and hydrophobic. They, like carbohydrates, are made of carbon, hydrogen, and oxygen. And the small units, or the monomer, is fatty acid. They act as long-term energy storage and structural building blocks, for instance, in the cell membrane, and also in things like cholesterols. Fats in our body are sometimes called triglycerides because they have a glyceride with 3 fatty acids dangling from it. Triglyceride. They store energy, and they can be unsaturated, which is something you've probably heard of before, or saturated. And that just refers to the bonds. If there's a double bond, it is a saturated fat. If there is no double bond, or if it is unsaturated, there is a double bond. And if it's saturated fat, there are no double bonds. If you ever want to check if you're eating saturated or unsaturated, just check the nutrition box in the back of your food. Steroids include cholesterol and certain types of hormones, and phospholipids is the lipid bilayer of this cell membrane. All right, so here's a good example of a difference between a saturated fat and an unsaturated fat. The double bond here causes a bend in the molecule. So if it's double bonded, it's going to be an unsaturated fat, whereas basically when we have a carbon chain without a single double bond, we're going to call that a saturated fat. Okay, the third one is proteins. Notice for proteins, carbon, hydrogen, oxygen, and nitrogen, sometimes sulfur. There's no phosphorus. It's composed of chains of amino acids. Proteins, I like to consider them the jack of all trades. They perform many functions in the body. They act as structural components. They help transport things. They act as enzymes. They signal cells and they do a lot more. Even our hair and a lot of like fingernails, for instance, are made out of proteins. Foods, we commonly find them in meat, soy, and nuts. And cells, we have a sodium to potassium protein, which helps it's a channel protein in the cell membrane. And then we have enzymes and cell signal proteins. In plants, some you might have heard of like latex. Then there's chlorophyll, auxins that cause plants to bend, and gibberellins. In animals, we have muscles, keratin, collagen, and antibodies, part of our immune system. So proteins do a lot, and they do. All right, this is a great example if you want to see specific examples, but I'm not going to go over this. All right, here we go. So proteins are made up of amino acids. The order of amino acids in a chain determines shape and function of the protein, just like it does with other chemicals. Amino acids have directionality. What I mean by this, if we look at the diagram, you notice there's an amino group, a carboxyl group, and a hydrogen. There's pretty much always going to be that. The R group, where it says variant, that's what changes. So depending on what it is, it gives the protein, or gives the amino acid direction, helps it connect to different things, get different properties like polarity and acidity, if it's an acid or a base. Just because it says amino acid does not mean they're all acids. In fact, most of the time, they're not acids. So the R group amino acid can have properties like hydrophobic, hydrophilic, or ionic that determine structure and function of that area of the protein. Here's examples showing what I mean by different R groups. Notice how we have our amine, hydrogen, and carboxyl group. And then every single amino acid, these are all the examples, there's 24 of them. I have 9 shown here. They are all examples of different R groups that provide, look at glycine, just one hydrogen compared to phenylalanine, which is basically an aromatic carbon ring. Aromatic, that's what the lines are. Okay, moving on. Last, we have nucleic acids. So nucleic acids have all the elements of the rainbow. They have carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. They're monomers, they are made up of things called nucleotides. You might hear nucleosides sometimes. We're not going to focus on that too much. Nucleotides are made up of sugar, a base, and a phosphate. Polymers of these nucleic acids include DNAs, RNAs, and then we have NADH, NADPH, and ATP, which is something we're going to spend a lot of time on, adenosine triphosphate. Amino acid sequences, which are known as polypeptide chains, which make up proteins, are programmed by a gene. Genes are made of DNA. So a nucleic acid is monomers of nucleotides, which means that DNA not only does it provide replication for its own, provided directions for its own replication, it also directs synthesis of messenger RNA, which is right here, mRNA, as well as protein synthesis, which is why you'll hear a lot of the times that DNA is the instructions for making every part of our body, because in a way it is the instructions for making the proteins. And since a majority of our body is proteins, that's not like water, that means it's the instructions for making our body. All right, nucleotide polymers. This is where the DNA synthesis can be confusing, but if you pay attention right now, I promise it won't be. So if you look at adjacent nucleotides, nucleotides that are next to each other. So the phosphorus, the sugar, and the base, that's one nucleotide. If you look at how it's organized, where the phosphorus connects to a sugar, you will notice that there's called a 3 and a 5 carbon. On top of headline, I've highlighted what I mean by this. So DNA runs opposite, 5 side to 3 side. One side of the DNA will be the 5 side pointing up, and the other side will be the 3 side pointing up. Notice how it makes it appear almost upside down. It gives it this off-level look. It's supposed to. The idea here is that with the 5 carbon on chemical molecules with a carbon skeleton, you can count the carbons. So if we look at this oxygen right here, we are going to count and see where it connects to the phosphorus group. 1, 2, 3. 3 end. 1, 2, 3, 4, 5. This carbon right here. 5 end. That is what we mean by 3 and 5 end. Notice how they run opposite to each other. In the middle, we have what's called a hydrogen bond. There we go again, which means that it's easy to separate than breaking apart any individual molecule and will allow easier times for DNA replication. Okay, here's a good comparison of DNA and RNA. The key I want you all to know about is this right here. deoxyribose sugar compared to ribose sugar. That has to do with a hydroxide group, an OH group, and that's something we're going to talk about in class as well, but I'm also going to mention it right now. The hydroxide group makes the difference. Okay, there's two things we have to talk about. Luckily, if you're kind of zoned out, don't worry, I'm almost done. Dehydration synthesis is a simple regard to using water and breaking apart a hydroxide group and a hydrogen to kind of link molecules. So dehydration synthesis will form a new molecule, and it's a way to make a monomer or two monomers into a polymer, or polymer into a longer polymer. So it creates a larger polymer. Dehydration synthesis gets its name because a water is created by removing a hydrogen from one and a hydroxide molecule from the other. In doing so, they form together and you get a water molecule that's removed, which dehydrates the two molecules, dehydration synthesis. The charges created from the removal of the molecules form a new covalent molecular bond demonstrated right here. There's your covalent bond, this line, where it used to be a hydrogen and hydroxide group on different molecules. Okay, here's a good example in biology. We have monosaccharides forming disaccharides. So we have glucose and fructose. See the different groups right here. That's what creates the difference in the molecule. Dehydration synthesis removes these two groups highlighted in red. You can see that here. In doing so, we have sucrose being created with an oxygen bond. Sucrose used in plant structure. Glucose, same thing happening, different form of structure because we're using glucose instead of glucose and fructose, forming maltose. This is a good example of dehydration synthesis in biology, and it's a demonstration of how glucose is used to form starches, glycogen, and cellulose. Dehydration synthesis removes water joined molecules together. It is an endergonic reaction, which means it needs energy. Anytime we build a molecule to create a bond, we require energy. Hydrolysis. Hydro means water, and we see that lysis, that means to break. So this is the opposite of dehydration synthesis. It's where we break molecules using water. So notice that this situation here, maltose to glucose and glucose, is the exact same as we have right here, but in reverse, honestly. Instead of taking away a water, we are adding a water, and in doing so, that allows us to break this oxygen bond by creating stronger attractions to it and separate the molecule into two glucose, just like we had before. So it adds water to break apart a molecule. We call this exergonic reaction, which means it gives off energy. We are breaking a bond. Bonds are energy. So when you break a bond, you create energy. When you make a bond, you need energy. All right, if I'm not mistaken, that is it. We have hydrolysis explained. You can go through this one on your own. Polymer is broken into two components. One part gains a hydrogen atom. The other gains a hydroxyl molecule from a split water molecule. All right, that is going to be it for the lecture. If you have any questions, please let me know and have a good rest of your day.